Alright guys, this is again Dr. Surasinghe with the lecture on internal environments. Particularly in this lecture we are going to talk about how um, animals regulate their internal environment in terms of biochemistry, in terms of temperature, and many other critical features. We are going to largely focus on the internal environment in terms of regulating concentration of minerals, nutrients, and organic food resources like glucose. Alrighty, so moving on. So first, when you're talking about regulating internal environment, I mentioned that we are going to concentrate a lot on nutrients. So let's talk about some basic topics, or sort of try to rekindle some of your basic knowledge from basic chemistry courses. What is osmosis? Osmosis refers to movement or diffusion of water, right? So you are looking at, um, in this beaker, right? You are actually looking at solution, right? A sugar solution, in fact, right? So, and then it is separated, and this beaker, in this beaker, the sugar solution is separated by a selectively permeable membrane, right? So on one side, on my left side, I'm looking at high concentration sugar solution. On my right side, I'm looking at diluted sugar solution, right? So on both sides of the selectively permeable membrane, we have two variable concentrations of sugar solutions, right? So diluted meaning what? Meaning there are only fewer dissolved sugar molecules are there per unit volume. On the other hand, there are more sugar molecules per unit volume. That's what that means. So the side with a greater concentration of the solute, right, is called hyperosmotic, right? And then the solution that has a diluted solute concentration with with fewer solutes per unit volume, that is hypoosmotic. So in the diluted side, in other words, there's more water potential. On the highly concentrated solute side, there is less water or less water potential. Okay, that's one. About the membrane, selectively permeable membrane, meaning it selectively permits certain molecules to move. In this particular case, it is selectively permeable for the movement of water, but not permeable for the movement of sugar molecules. Because of that, with time, what's going to happen is water molecules is going to actually move from the diluted sugars solution to more concentrated sugar solution. Why? Because there's a greater water potential on the diluted sugar solution, less of water potential in the concentrated sugar solution. So water will move from high water potential to low water potential. In other words, water will move from low solute concentration or from low sugar environment to high sugar concentration environment. So that's actually that kind of movement of water along the concentration gradient, from high solute concentration, from low solute concentration to high solute concentration, is actually osmosis, right? So why sugar molecules are not moving? Because the permeable membrane does not allow sugar molecules to move. So again, what I want you to understand is what osmosis is. Movement of water, right, from where there's a lot of water available to areas where there is less water available, right? For that movement, whatever the barrier in between should be permeable for water. Okay. And again, in another way to word osmosis is movement of water from hypoosmotic side to hyperosmotic side, right? So that's actually how this happens. Now, this happens biologically, right? This is actually kind of the more conceptual diagram. Net water flow from hypoosmotic to hyperosmotic side, right? From low solute concentration to high solute concentration side. Okay, now that happens at cellular level, right? So this selectively permeable membrane exists biologically. The cell membranes are actually permeable to several minerals as well as to water, right? So water can actually pass through cellular membranes. So if you take a red blood cell or a sample of red blood cells, and if you were to put it in water, right, in like regular drinking water, right, what would you expect to happen? Well, in fact, the red blood cell, if you put it into like a tap water, right, or for drinking water, the red blood cell inside contains lot of solutes compared to the outside, right? So as a result, what's going to happen is water will move from the surrounding into the red blood cell, and the red blood cell will start to expand, and ultimately what happens is the expansion has no limits. It will expand until the cell membrane ruptures, right? So so that's what actually shown by this particular diagram. Here you can see when you place a red blood cell in hypotonic medium, right, we are going to observe water moving into the red blood cell from the surrounding environment. Why? Because in the hypotonic solution, the hypotonic medium, right, in our case drinking water or tap water, has less solutes than inside of the red blood cell. So water moves into the cell, and it actually, so the red blood cell expands. Ultimately the cell membrane cannot hold the cells together, it ruptures. Okay, so that's actually one situation. The other opposite situation is what happens if you put the red blood cell in high concentrated saline solution? Well, the highly concentrated saline solution, if that saline solution happen to have a greater solute concentration than inside the red blood cell, you are looking at a hypertonic medium, right? So in that situation, water will move from low solute concentration red blood cells to high solute concentration medium, the hypertonic medium. So as a result, the red blood cell will start to shrink, right? Ultimately, it will end up like a clot, like you see in this image. If you place the red blood cell in mammalian saline solution, where the solute concentration is comparable to that of inside the red blood cell, you will not see any net movement of water, right? So as a result, the red blood cell will not lose its shape, or its it will remain intact. It will not shrink, nor it will rupture ultimately, right? So so that's actually something I want to explain. Now, for example, if you synthesize blood plasma, right, with which you can do, it will be hard, but you can do, and if you put a red blood cell inside that plasma, you will experience the same situation as here, right, as what you see in a hypotonic medium. Okay, let's go. Now let's try to relate that concept into real life situation, right? So in certain environments, right, you can actually so as an organism, depending on the environment you live in, you need to regulate your internal body conditions. There are two ways animals can achieve this target, right? Your environmental stress is the environment you live in, or the medium you live in. Let's say for a fish example, right? For aquatic organisms, this is actually rather drastic than for terrestrial animals. If you are an aquatic animal, like a fish, you either need to regulate your own osmotic potential or your own solute concentration with respect to the external environment, or actively regulate or mitigate gain or loss of water or gain or loss of solutes, right? So those are the two options you have in terms of countering the stress that is coming from osmosis, right? Or the stress that emerging because of different solute concentration in between inside the animal's body and the animal's environment. So there are two ways animals can deal with this situation: osmoconformers and osmoregulators. Now remember again what osmosis is: movement of water following a solute gradient, right? So following a solute concentration gradient. So that's what, so osmo refers to the osmotic stress or osmosis induced stress. Conformers is kind of agreeing, right? So many marine animals are osmoconformers. What does that mean? There are many marine animals that do not actively regulate osmosis, right? What they do is they can tolerate a huge variation of internal solute concentration. So they will either, depending on the environment they live, they will either permit solute movements into their body, or they will permit solute movement out of their body, right? They will also permit either water absorption or water loss or dehydration. So if a particular animal does not actively try to regulate their internal body chemistry, right, internal water levels, right, you are looking at an osmoconformers. So their internal solute concentration, their internal water potential, internal levels of water will fluctuate based on the environment they live in. So because they don't try to regulate their internal environment, their solute concentrations, in their body fluids inside their body will always equilibrate with that of the environment. Because of that, we call them isoosmotic, meaning their internal environment will always vary to an equilibrium level, to a similar level with that of the environment. Okay, so they are not actively attempting to regulate their solute concentration. That's what I meant by do not regulate osmolarity. They don't try to regulate their solute concentration. Okay, osmoregulators is actually the part where animals energetically, right, attempt to control water uptake, attempt to control solute uptake, right, or attempt to control either uptake or loss, right? So if the animal, so in this case, right, the animal's internal chemical environment, solute concentration, right, will stay constant. It will not change with respect to the environment, right? So if you can look at if this kind of animals who can regulate their internal body chemistry, right, will hold their internal body chemistry constant, right, whether they are on a hyperosmotic environment or hypoosmotic environment. Okay, so let's take a look at a picture to understand this, right? A graph to understand the differences between osmoconformers and osmoregulators. This is something actually what I do in a real class. I ask a question, but right now I cannot ask the question. So x-axis is external solute concentration, right? And y-axis is the internal solute concentration, right? So this particular line marked B, right, it actually corresponds to an osmoconformer. So as you can see in the osmoconformer, their internal solute concentration vary the concentration of the external environment, right? Now look at the line A, right? That's an osmoregulator, right? Osmoregulators' internal solute concentration does not change, right? So the so they can occupy a wide variety of external environments without no significant change in their internal solute concentrations. So that's actually the difference between osmoconformers and osmoregulators. So there we go. A is osmoregulators, B is osmoconformers. So there is going to be kind of a variability. Like, I mean, it's not, you cannot always put a particular animal, as a textbook regulator or as a textbook osmoconformer, right? Yes, of course, some animals, like you see in this purple line, they are strict osmoconformers, right? The degree to which their internal body chemistry conform to the environment is perfect, right? I mean, you can see it actually happens very close to the 1-to-1 ratio. Okay, now over here you have a strong regulator, right? You can see actually earlier, right, there's solute concentration changes, internal solute concentration changes with respect to the environment, but after a while it becomes stable and remains stable, right? So this is kind of a very strong regulator, right? Very strong osmoregulator. Well, some of them actually are not that strong, right? They actually earlier, what happens over here, you know, slowly the body osmotic potential or the body solute concentration increases, then reaches stability for a shorter while, right? It remains, the internal fluid concentration remains stable with respect to changing environmental solute concentrations, but after the environmental concentration exceeds a particular level, the animal loses its ability to regulate and becomes a conformer, right? So there you are looking at a weak regulator, right? And here you are looking at a perfect textbook osmoregulator. There is no change in the internal body chemistry with respect to the external environment, right? That's kind of a textbook osmoregulator, right? In real life, you don't get perfect osmoregulators, you know, because animals or regulatory systems are not that perfect. And then some of them actually can, you know, fluctuate between, you know, like over here in this guy, right, in this particular animal in orange, you are looking at, you know, earlier it's able to create some osmoregulation, right, but after a, you know, a significant tolerance of osmoregulation, it loses ability to regulate and becomes an osmoconformer. So some animals can actually sort of jump in between osmoregulation and osmoconformation, right? So it's actually very interesting that in real world things are a little bit complicated. Okay, now let's remember what I talked to you about the animal kingdom. During the evolution of the animal kingdom, they actually moved from water to land. So when you adopt a terrestrial mode of life, and as an animal, you need to evolve strategies to minimize dehydration and desiccation, right? So that's actually a very important part of osmoregulation, which actually enhances the need for osmoregulation, particularly in terrestrial animals. And that does exist in freshwater animals as well, because especially in a marine organism, marine animal, you have to prevent dehydration too. So homeostasis, it's a term that I introduced to you before, but I'm taking that term back here. Homeostasis is a physiological, behavioral mechanism that animals use to regulate their internal biological conditions. Through a complex system of feedback biological mechanisms, these animals sense what's happening in the external environment, and the feedback mechanism sort of regulates the internal conditions and help keep the internal conditions in rather constant uniform level. Okay, and so thereby these the animals that can perform homeostasis can stabilize their optimal biological functions, right, and then maintain their internal biochemical activities, physiological activities in the optimal health condition. And that can happen regardless what's going on in the external environment. So osmoregulation is a form of homeostasis, right? Now in osmoregulation, what's happening is those animals attempt to maintain a relatively constant optimal internal osmotic pressure, right? How? By having a stasis, having stability in terms of body fluids, gaining stability in terms of body solutes, and maintaining a suitable concentration of solutes and water inside tissues, inside cells, right, in the extracellular space. And thereby, because these animals maintain a constant levels of water inside the cell as well as in between cells, as well as generally in their living biological bodies, they can maintain their physiological activities in optimal level, right? Because physiological activities are basically what? It's metabolic, right? It's all physiological activities are different types of metabolic activities, right? And metabolic activities are, there's an optimal metabolism, and then anything below that is suboptimal. Anything above that is also not optimal, right? So if these animals are able to maintain regularity in terms of solute concentrations and water level, right, it facilitates the maintenance of optimal metabolism, right? And also to maintain optimum circulation, that's also important. Why? Because circulation, the efficiency of circulation, I mean, we learned about circulatory system in the previous lecture, right? And I did mention that the blood plasma is largely made out of water, right? The transportation mechanism largely is based on presence of water in blood, right? And also the communication between cells, right, is also dependent on the presence of optimal level of water, right? So if you do not protect water inside the living organism, you will actually have problems with your circulation, right? Literally, like, I mean, if you are dehydrating a lot, you are actually losing the viscosity of your blood plasma, right? So your blood will not flow properly. If blood doesn't flow properly, if it is too thick, right, then that actually hinders transportation of matter and energy. The same thing and how does the animal keep this kind of osmotic potential? There are largely there are multiple mechanisms, and to the greater degree, maintaining optimal solute concentration and water levels inside an animal's body is either maintained by chemicals, that is endocrinal function with hormones, or neurological by involvement of the nervous system. So let's try to understand this homeostasis, right, with respect to this particular diagram, right? So internal body, right, because of changes in the environment, the internal body initially is subjected to change, right? So because of that change, right, what you observe is the body condition changes from this set point. The set point is the optimal level, the best ideal level, the textbook level, right? So if the internal body conditions deviate from this set point, if it changes from this set point, that change can be detected by a particular sensor, right? Some sort of a biological sensor inside your body, some sort of a sense organ will detect that shift in your solute concentrations, right? And then that sensor will actually communicate through different other organs to result in corrective mechanisms, to elicit corrective actions, right? So there will be several corrective actions happening through other body systems, right? Those body systems could involve your endocrine system or your nervous system, right? And those so the integrator, right, what sensor triggers is an integrator. Your integrator is going to be in the nervous system or the endocrine system to the larger extent, right? And then whatever does by the integrator, your nervous system or endocrine system will trigger a particular effector, right? Another organ that actually is responding to changes in the nervous system or changes in the endocrine system, right? So for ultimately what happens is, you get a corrective mechanism triggered. That corrective mechanism reverses back the changed internal condition back to the set point, back to the optimal level, right? And once your body internal environment returns to the set point, to the optimal level, right, then it will again trigger your sensor, right? And the sensor then will actually turn itself off, because why? Okay, the set point is reached, there's no point of sensor triggering any more actions, so sensor turns off, so corrective mechanisms then shuts down. So meaning what? The integrator in this case, right, the nervous system or the endocrine system, they will stop stimulating the effector, right? So at a very conceptual level, this is how homeostasis happens. So, well, how does the endocrine system or the nervous system, right? Those are your integrators. How exactly do they trigger an effector, and what would an effector does? What can be an effector? So with respect to osmotic balance maintenance, right, which is actually one of the homeostasis I'm going to concentrate on, it's largely mediated by physiology, right? And the physiological part is mediated by hormones, like I mentioned, right? So certain hormones could actually trigger many physical reactions, right? It could trigger many systems in your body, right? So there are other mechanisms where osmotic balance can be maintained, right? Like how? Behavioral mechanisms, right? The effector could be behavior. In fact, some of the neurological integrators, right? Certain neurologic systems can actually change your behavior, right? For example, drinking water when you are thirsty is a behavioral mechanism that is neurologically modulated to keep the solute concentration or water balance in your body. Or eating food in the animal world, when animals are thirsty, well, of course they too go in search for water and drink water. Sometimes they also eat food that are rich in moisture level. That's a behavioral mechanism triggered neurologically, and to a certain level, that kind of behavior of searching water or eating moisture laden food can also be triggered by hormones. It's really interesting even to think about that. And morphology and anatomy. Now, certain adaptive features, right, that are actually rather non-reversible, or rather takes a longer time to change, can also happen through morphological and anatomical means. I'm not going to go into details of those anatomical and morphological adaptations. Rather, I'm going to largely concentrate on physiological ones. But do recall that is also possible. For example, animals that live in dry environments like deserts, right, they have to prevent dehydration. They have to prevent water loss through evaporation. So these animals in desert environments, like particularly reptiles, have a very thick skin that is impermeable to water. So that's a morphological and anatomical adaptation, having a skin that prevents water loss, right? And to a certain degree, there are developmental biological adaptations where the adaptation is observed during the embryonic developmental stages. Now, that is something I am not going to go into any detail because it's too much to cover in this level in a primary biology course. So now our next task is to try to go by step into how internal environment is regulated. But this kind of internal regulations in an internal biological environment, right, that regulation is largely driven by cell-to-cell signaling. That's how the nervous system communicates, right? It's a mechanism. Nervous system is a very interesting system which is well adapted to fast cell-to-cell communication, right? The same thing regarding endocrine system as well. So the cell-to-cell signaling is really important to be stimulated by the external environments and then create a trigger to trigger a response to stabilize the internal environments through nervous system or through endocrine system. So in terms of the nervous system, right, how nerve cells communicate, how neurons communicate is by conducting electric impulses, right, along a cell, and then of course jumping from that cell to another, right, by using chemical signatures, right? That's actually basically how nerve impulses are conducted. Within a neuron, it passes through the neuron as an electric signal, literally an electric signal, right, electric current, and then how it jumps from one neuron to another is by secreting a particular type of a chemical from one neuron, which migrates towards the other neuron and kind of makes the signal more rather continuous. And so chemical signals that we observe in animals, those chemical concentrations be quite low. Despite the lower concentration, those chemicals can elicit an enormous, large response in the entire animal, right? So the low concentration of certain biochemicals does not mean that they are not doing any function, right? What means is even at lower concentrations, that chemical can have a greater impact on that animal. So the last point I want to mention in terms of cell-to-cell signaling is that the nervous system largely uses electric impulses, right, to send the signals. And if you look at the endocrine system, that's largely based on chemicals. Now, both systems are equally important. So because nervous system largely depends on electric impulses to conduct signals, nervous systems can elicit a short-term response, right? So the response is elicited really fast, right? And the response is short-lived, right? But in contrast, chemical signals that are mostly observed in the endocrine system, those responses takes a long time to take effect, right? Endocrine system requires relatively longer time span to respond, but that respond will last longer, right? Again, nervous system, short-term responses, right? Quick and rapid responses. Endocrine or rather chemical-based systems, it takes longer time to elicit the response, but the response lasts longer. Okay, let's move on. So, there are many different categories of chemical signals that are important to regulate the internal environment. I'm going to go over 5 basic categories of chemical signals that I want you to know that are important for homeostasis in animals. First type is autocrine, right? Autocrine signals are produced. In case of autocrine signals, the chemicals are produced by a particular cell, and that chemical elicits a response on the same cell that produced it, right? So like, look at the diagram, right? It is produced by this particular cell, right? And the impact of that produced cell is on the source itself. So the source of production and the target of that particular chemical is the same cell or the same organ, right? So the example is things like a cytokine, right, also known as interleukin 2. It's actually a chemical produced by T cells. T cell is one of the immune cell in your blood that triggers the T cells to fight better against infections, right? So T cells produce cytokines, and once produced, right, the T cells become better equipped to fight diseases. Okay, the second type is paracrine signals. Paracrine signals, when produced they are produced by a particular cell and then diffused locally into the other cells or other organs, nearby organs, and elicit a response in closer by cells or closer by organs. Now the examples are insulin and glucagon, right? Those are secreted by certain cells in the pancreas, right? And then they actually act like paracrine signals and eliciting the response to regulate glucose concentrations, right? So insulin, what it triggers is it triggers taking up blood glucose and it triggers absorption of the glucose. Glucagon helps the reverse, right? Glucagon can actually induce the body to release glucose into the bloodstream. So now I know that insulin and glucagon are also hormones, but they also have a paracrine function, because when they are produced by the pancreas, they could elicit a response in the nearby cells, not just cells that are further away, not just the organs that are further away. Endocrine, also known as hormones. Endocrine signals are also known as hormones. So these categories may not be necessarily mutually exclusive, okay? Because now already you know that endocrine and paracrine have some sort of similarities. Endocrine signals, by definition, they are hormones. So those hormones or those endocrine chemicals are produced by one type of a cell, distributed to a different target via body fluids or by blood, right? So produced and secreted by specialized cells, right? And then transported to a different organ, different types of cells, different tissues to elicit a particular response, right? The source and the targets are not the same, and nor are they close, right? They are further apart. So you need a circulatory system to transport endocrine signals. Another part, the fourth type, is neural signals. Now, in neural signals, the chemicals are produced by neurons or nerve cells, and that produced chemical signals travel a very short distance between nerve cells, right? So the example is neurotransmitters like acetylcholine or dopamine, right? That actually is produced by a particular neuron, travels through the synapses, the interneuron gap, right, between neuron gap, and then triggers, creation of another electric impulse in the other neuron. So and then the last one is neuroendocrinal, which is kind of a blend between neural signals and endocrine signals, right? So these are neuroendocrine signals are chemicals produced by neurons that are secreted into the bloodstream, right? And then transported into a different target and elicited the response in the target, right? Well, those are the basic types of chemicals that I want you to understand. But again, I want to emphasize that those are not mutually exclusive, right? So for example, there's a very strong similarity between endocrine and neuroendocrine. So only difference between endocrine and neuroendocrine is when it comes to neuroendocrine signals, the source of the chemical is a neuron, right? In endocrine signals, usually we are looking at a non-neuron cell that is producing the chemical, right? So and the difference between neural signals and neuroendocrine is that in neural signals, the chemicals travel very short distances, and usually it's from one neuron to another, right? And but on the other hand, in the neuroendocrine, it could be produced by a neuron, and it could elicit a response in any other target cell, target tissue, or a target organ. So there you go. So what I want you to understand is actually kind of have an appreciation for these different types of cell signaling and have some understanding about an example for those, right? Largely the difference in this case is actually the source of the signal production and what the target is, and whether the target and the source are closer by or further apart, right? So those are basically the subtle distinctions between these 5 types I just explained. Now this lecture about internal environment is going to concentrate quite a lot on hormones. So what is a hormone, right? We have actually kind of gotten a good approach to talk about hormones thus far. So if you were to define a hormone, these are the 3 critical ingredients that you need to remember. Affects distant targets. When a hormone is produced by a particular source cell or a source organ, right, the impact is actually going to be going to last for a greater distance in the body, right? That the target of that particular hormone is not the same as the site of production, right? Most often the hormone travels some distance from the source towards a particular target. So if there's a distance gap, a kind of a physical gap between the source cell and the target cell, right? How would the chemical travel? Well, either largely by some sort of a body fluid, right? And it could be, in case of mammals or in case of vertebrates, that's going to be largely blood, but it is not always blood. Sometimes many tissue fluids, this interstitial fluid, the water-based viscous medium that you find in tissues, can also act as a medium to mobilize hormones from the source of production, from the source to the target. So that's the second part, the travel component, right? The third component is actually the response it elicits. So it actually is produced by a particular source cell or a source organ, mobilized through interstitial fluid or body fluid or through blood, and elicits a response at the target. right? So every hormone is designed to elicit a particular response from a target or multiple targets, right? And so the response elicited is remarkable, right? The hormones usually are produced in lower concentrations, in tiny amounts, small amounts, but the response elicited is not insignificant, is not trivial, right? The response is major, right? It's extremely significant, life-saving, right? So those are the three major components, right? Produced by a particular cell or an organ system, right? And it elicits a response from a distant target cell, distant target organ system, and travels from the source to the target through a particular a fluidic medium, right? And elicits a response that is significantly greater compared to the concentration of the hormone. Okay, hormones vary in terms of the source and the target, right? I just mentioned that several times. So some of these hormones, the distance they travel is minimum, right? Sometimes the elicit, the target or the response elicited is rather local, right? From the source, it diffuses locally towards local targets and elicits a particular response. In certain cases, it could be long distance, long ranged, right? It could be produced by a particular gland, as a chemical secreted into blood, and take some time even for the diffusion to take place and to elicit a response, right? I'm talking about within the same organism's body, but still there's a distance to travel. And sometimes this long distance travel is also mediated by neurons, right? So it could be a neuron or neurological organ that is secreting the enzyme, secreting the hormone, my bad, and that hormone is then produced or released into blood, and then it's mobilized towards a particular target cell. And in certain cases, some of those chemicals produced, right, left a particular animal's body, right, and it is left in the environment and mobilized through the environment to land on a target animal, right? Pheromones is an example, right? So in certain, some biologists consider pheromone as a different form of a chemical from hormone, but if you look at the biochemistry and how pheromones function, I would rather consider pheromones as an exceptional type of hormones. Because the reason I consider that is in certain social animals, elephants, for example, hormones released by a particular individual elephant could elicit a response in other elephants of the same group, particularly in female herds. This is quite remarkable, right? You may have heard that when a particular wild elephant is producing milk, when she has a baby, all the females in that particular group starts producing milk. That's because the hormone secreted by the actual biological mom elicits a response not just from the mom to produce milk, but all the other moms in that particular elephant herd. So because of that, I wanted to consider pheromones, the fact that certain hormones can function like a pheromone, right? Where hormone is produced by a particular animal, released to the environment, and elicits a response from another individual, right? Sex pheromones also has a similar effect, right? Certain pheromones, how certain males attract females, or how certain females attract males, is by actually releasing pheromones. That is actually where the producer releases the pheromone to the environment and then lands on a targeted individual that elicits a response. Kind of really cool when you think about that kind of stuff. So like I mentioned very early, the cell-to-cell communication in terms of chemical signaling is quite important to understand, right? So these chemical messages, hormones, are released by a particular source cell or a source organ, right? It could be a neuron or a nerve cell, or some sort of a gland or a glandular cell that is releasing this particular chemical, right? And then, after traveling through body fluids or whatnot, the signal is then intercepted by a receptor protein on the target, right? So in this case, this is the chemical signal, right? This is some sort of a molecule, right? In lower concentrations, that is traveling through the body fluid, right? And this part is actually the target cell, right, or the target organ, right? And this particular protein is a membrane protein embedded on the plasma membrane, right? And that protein is a receptor protein. That protein is specifically designed, right, through evolution, specifically designed to bond with this signal molecule, right, the hormone that is kind of coming through the body fluids, right? So upon this receptor protein bonding with the signal chemical, right, it actually undergoes something called a conformation change, or in other words, a shape change in this particular protein, right? And that shape change, or the conformation change, relays a signal from the plasma membrane towards the cell interior, right? So what's happening over here is actually that, right? This conformation change in the receptor triggers this signal transduction pathway. It's kind of a way cells when they are stimulated at the surface, that's how they actually communicate that towards the cell interior, right? So this signal transduction pathway, don't be intimidated by that particular term. It means signals traveling through the cell, right? It actually could involve multiple proteins, right, or certain structures released from this receptor protein physically traveling through the cytoplasm, right, or the cytosol, right? And ultimately it will after several steps it will actually activate, right, a particular response or deactivate an already existing chemical reaction or a cellular action, right? So this chemical signal or this relayed signal will either activate or initiate something or deactivate something that has been already happening, right? So this could be actually, could involve so many things. You know, we heard about gene expression, right? This kind of a triggering could actually trigger, this kind of triggering will actually lead into gene expression. It could, this signal transduction could go from cell membrane all the way to the nucleus, right? And or it could be releasing an enzyme from the cell, right? Maybe releasing the enzyme to the outside, maybe releasing the enzyme inside of the cell, right? And it could also trigger some of the other surface protein, like, you know, channel proteins, to open or shut and permit movement of minerals or ions. So the activation or deactivation could be many, right? So it could be, if one of those protein channels are open, right, this particular process could actually trigger the channel to shut down, right? Or if a particular enzyme is already being released by a cell, this kind of a mechanism could also stop that enzyme from being released, right? So there are so it could be an activation or activation of a process, triggering of a process, or deactivating an existing process. Let's move on. So again, what is a hormone now in terms of chemically speaking? Well, hormones could actually have these three sort of chemical formations. First type is polypeptides, right? The example is oxytocin, right? And just to give you an idea what is oxytocin, what it does, one of the few critical functions of oxytocin is in female body, particularly to trigger contractions of the womb for child delivery, for childbirth, and then for lactation, right? And then so oxytocin is a polypeptide, right? And what is a polypeptide? Think about like in the process of protein synthesis, when you have multiple chains of amino acids, you know, bonded together, you could actually have a polypeptide, right? And then you have many polypeptides arranged together in a particular conformation, you get proteins. And some hormones are actually more complex proteins, like growth hormone. What does growth hormone do? Literally responsible for your physical growth, enlargement of your muscles, growth of your bones, growth of your entire body is driven by the growth hormone. Some of the hormones are actually amino acid derivatives. Two examples, thyroxine and melatonin. Melatonin is an interesting hormone because that's actually the hormone that triggers your sleep-wake cycle, kind of creates a behavioral modulation based on the day and night, Thyroxine, it's actually a hormone that is important to regulate digestion, proper function of heart and the and skeletal muscles, brain development, and the maintenance of bone structure. And third part, I think this is the part that you guys are largely familiar with, lipid-based hormones that are called steroids, right? Most of the sex hormones that you can think of, right? Estrogen, progesterone, testosterone, those are actually the lipid-soluble steroid hormones. Now it's interesting to think like how these different types of chemicals communicate between cells, right? If it is lipid based or lipid soluble or steroid based, then that kind of hormones can actually enter the cell through the cytoplasm, through the cell membrane, because cell membrane has a lipid bilayer, so they can go through the lipid bilayer. But other hormones like protein-based one, they actually have to communicate with these receptor proteins to enter the cell. It's kind of interesting. Now, receptor proteins that are found on cell membranes, right, can actually respond to more than one hormone, right? So the actual function of a hormone cannot be solely predicted by the hormone's chemical structure or its molecular arrangement, right? It's actually it's a combination of the hormone and how that hormone communicates with the receptor proteins collectively, right? That determines the function of the hormone. All right, so a particular receptor can actually respond to multiple types of hormones. And there are many organs in your body, right? Many different glands and neurons that can produce different types of hormones that have different chemical structures, right? So this is why if you read, your textbook, like what functions hormones do, you might think, holy crap, like, this hormone does this and that. Like, I mean, remember what I mentioned about thyroxine? Digestion, heart and muscle function, brain development, bone maintenance, right? They are not even related functions. That is simply because a particular hormone, right, can trigger different cascading mechanisms, cellular mechanisms, by triggering different types of receptors, and a single receptor can actually elicit multiple responses to different hormones. So it's actually, this is why, if you wondered why chemicals are so bizarre in terms of their function, that's because actually they are designed evolutionary to function that way, right? The image on the right shows you different locations, anatomical locations of different organs that have endocrine functions that produce hormones. Some of them are actually located in your central nervous system, right, embedded in your brain, right? And some of them are like thyroid, like it's on your throat area. And there are many endocrine functions in the digestive system and the glands associated with the digestive system. Stomach itself can act like an endocrine organ that produce hormones that can regulate digestion and absorption, right? And then those adrenal glands found on top of kidneys. Kidneys themselves can produce hormones, right? And then the sex organs can produce hormones. So hormone production happens all over the body, can happen in many different organs, have that completely different functions. Okay, this is actually end of the lecture on the basic introduction to hormones. Then we will follow up with different examples of different hormones and how they elicit certain responses to maintain constant internal environmental conditions. Okay, guys, talk to you soon.